I'm going to present IDR that was mentioned in the previous talk by Matthew. So one thing already to keep in mind, IDR is based on Omero. Omero was mentioned as well. So everything you would see that's Omero under the hood, basically. So IDR is public access and it's about reference data sets. So Matthew mentioned BIA and Empire. Here we'll focus on a subset of some study, and it's highly curated. So that's one thing to keep in mind compared to other repository. There is a curation phase that is quite important. It's an important factor. As it stands, we almost host around 200 terabytes. So we did a release a few days ago and covered around over 80 organisms. So it's a large number of organisms. We host gene and antibodies and compounds. So that's where the curation part is important to keep in mind. So how does it look? So when you look at submitting data, so we are working, as Matthew indicated, in collaboration with the Bioimage archive and also with Empire. So really depend of what type of data you're submitting will direct you to the correct location, because IDR will only take a certain subset. If we estimate that there is, for example, a new organism that we just published a study about biodiversity that contained 14 different organisms that just got published. So this is the kind of, it's a decision that is made when the author contacts us for data submission. So the process is not a form to fill, because you can't. The first step is to contact the IDR team. We assess, we submit the raw data. We assess the file, and after there's template to fill, and the template. I mean, filling a lot of information about ontology and metadata associated to the study. So it can be potentially a longer process than just uploading a file and be done with it, because we require, in a sense, the richness of the metadata. It's an important factor to keep in mind. So a publication from contact to publishing could take months, depending of the type of curation required, and also the submission process, because at times we realize people have lost file along the way. And to support we support all the file format that bioformat support, because I said it's Omero under the hood, so it's around 165 format. And there were questions earlier on about the type of data. We support anything that bioformat supports. So that go from human data to ocean data to cell to different, anything that is kind of deemed of value and curated, that can be curated and that represent a reference data set, basically. So I think I was trying to, in that talk, also present, because Isabel presented all the FAIR principle and how we try to be fair. I think that the main message is, why should you also spend a lot of time curating your data? What are you going to do with this type? Why is it useful to curate that data, and what can you do with it? So I was trying to connect the FAIR principle to its implementation. And again, it's trying to be fair, because there's nothing that is, as far as I've been involved in the kind of verification of data, I've not come across something that is 100% fair. It's a journey. And then ourselves, we continue that journey, because we add support for, Matthew presented the REMB and around earlier on about ontology that can be used for data acquisition, something we didn't look in the first place. Then we are now looking into. We're focusing more around gene and phenotype and compound, and now we need to focus on other elements as well in terms of curation. So the first letter of the FAIR is trying to make it findable. So who are the author, about license, keyword, parameter, and about what can we capture about the data that is relevant. So first thing is we link the data to the publication to the original data. So you will have a publication DOI. The data will also have a DOI, and we kind of there is already a linkage between the two. in a sense similar to what we were doing in the breakout session with the exercise of Zenodo. You had the DOI for the data and a DOI for the publication itself. And it's kind of this similar approach here. Here you're looking at some IDR published data about phenotypic trait diversity, but the important bit is on the right-hand panel. So you have data about the author, the PubMed identifier, publication DOI, the license that we use, the data DOI. So all those information we capture and we link to the study. So we store that information, and when it's possible, we make link to external resources. And you also can see there the biostudy accession numbers. So that's the collaboration I was mentioning with some. So the data go in IDR and also in BI, and there's linkage between the two. And if there is a publication, it's also accessible in the reference data set that is also published in Empire, the resources get linked. So it's not a disjointed effort in terms of the data, those data repository. Accessible. So make the metadata accessible, so you can be web UI. So we have a web front end, web viewer. You can, I would encourage you to browse data at idr.openmicroscopy.org. And an API access. So that's more the programmatic side of things that people want to do when they want to do AI or any harvesting of data in a larger scale, because human can have only a certain number of hours in their day to do certain things. So here you can look at some data and search for some gene and find this information. So that's kind of the web front end of the metadata. So those are, because data have been highly curated, that we're able to find some gene symbol associated to some images, and from there you will get the images. And you can find, for example, those are from the Protein Atlas, and you can find all those images and browse through them and access the data, visualize them in the web, and do anything that you will do with normal visualization tool. Here, I think Katrina was mentioning the Terra Ocean. So that's the interesting part here for me was just the annotation part that we spend time curation, the provenance. Katrina indicated in the chat earlier on. So you have the provenance. So here, the Terra Ocean project, they acquire a lot of images throughout the globe, put information about where that images was acquired, its location on the globe. And that pieces of information is associated to the image, which then allowed us to programmatically harvest that information. So here we are now using the API part, the accessible part with an API, and which allowed you to plot. So this is a Jupyter notebook where you actually plot where those images were acquired on the globe. And if you do it in an interactive manner, if you click on one of those red dots, you can actually see the image. So that pieces of information, the why you will require all this type of information to be stored along this image is then after that you get a meaning to from just a collection of points. You can actually think that image was in kind of acquired in South America, for example. So if we look now at the third letter of the FAIR approach, again, going back to why do we curate, is using, so it's the human-machine interaction. That was a point we discussed a bit in the group chat earlier on, is yes, I can go back to the paper and find that information, but a machine most of the time will not be able to do that. So how can we make it possible to find those images or those volume of data quicker? And that's a lot coming down to the usage of ontology. So in IDR, we use several ontology depending of the part we want to curate. So for gene, we use Ansible. So Ansible, and that usage of ontology afterwards allowed us we link those terms to the image data. So that's where I was saying the publication process can potentially take a bit longer because the information need to be provided. At the moment, there is a lack of tool for automation. You know, you can have yes, we can take, if people have already some Excel spreadsheet, we can parse the content, but I would say there is no automated weapon to say, okay, give me anything and I'm going to fix and find all the termini ontology for you. That's not existing at the moment. So it's a lot of man hour, and my colleague Frances Wong, I've seen her attending, is doing a lot of that work, and she can probably indicate some of the pain she has to go through at times. So, but the important thing is you have associated to a phenotype, you have a gene symbol, so you can then from that gene symbol also find other studies that you cannot not aware of. So this is kind of the discovery aspect. It said, okay, the why should you spend a lot of time curating? Because now somebody looking for some data, we're talking about AI or scripting, can then find other study that they didn't know about because they have associated to either a phenotype or gene, can then find other data associated in the register, in the repository, that have also that gene associated to the data. And you can see next to the phenotype accession, there's a logo. So that's EBI, so the ontology logo, in the sense that if you want to know more about that ontological term, you can actually find that ontology. So everything, all the pieces from those resources start to be connected one to one another. So the last letter is to be reusable. And one of the things is, well, the licensing issue was mentioned by Isabel. So we use a lot Creative Commons CC BY. We have CC BY license on most of the study, I would think. So we allowed people to reanalyze the data that are published in IDR, and that's one of the last part of the FAIR process, is making this data reusable. Again, it's another incentive for enriching your data, because someone which has nothing necessarily to do with that part of the research you were doing can find that pieces of information and do some further research. So here is an example of the COVID data set, so COVID-19 data set that was published in IDR during the COVID pandemic, where the eOpen Screen team was looking at numerous compounds, around 300 compounds that could have an effect on the COVID-19. So associated to that study, we work with the authors. They have done some scripting analytical tool, and so that was done in R, and then they were keen on showing the feasibility of that effort. So we have created a set of Jupyter notebook that we can link to the data, that we allow us to explore the result that we're presenting into a dynamic manner. So this is just an example of an example notebook. So they were calculating the IC50 factor for the compound in the paper. So in the paper, they indicated that remdesivir was the best and had an IC50 of 0.76. And when we're doing recalculation, so we were at 0.8. So you have always a fluctuation factor, error factor. So you can actually see the benefit of submitting not just the data, but all the information associated to it. So a user can come to IDR and change and decide it, because that is quite dynamic. Instead of saying, I don't want remdesivir, I want another compound. As I said, they were using 300 different compounds, and they can actually replot the result and find the information. So they can plot the dose response. So for remdesivir, you get exactly what presented in the paper. If you select a different compound, you would have a different result. So you have kind of very interactive manner of exploring the data. So that's by making the result reusable. And interestingly enough, as part of the EOS Life effort, you can also get recognition for that work because those analytical workflow can be published onto the workflow hub. And now you have a DOI associated to the workflow. So if you take the chain of submission, we have a DOI for the original publication, we have a DOI for the data, and a DOI for the analytical result. And you can actually have a body of recognition along the way, because a lot of the time people doing the image analysis do not have the recognition they deserve on that. How am I doing on time, Isabel? Am I over or still a bit of time? Or?
Yeah, if you could quickly wrap up, that would be nice, so that we have enough time for the last
yes,
session.
of course. Yes. So this is maybe another example done by. So this is a genome sequencing data set that can have a lot of DNA sequence in each individual cell. So that was part of the 4D nucleome project. So all the data are stored in IDR and with a lot of metadata. Ourselves, we didn't do anything with that metadata, but there is the project called the 4D nucleome browser that looked at those data and associated to those, as I said, the DNA sequence. They can access the image and link the image to this little dot I was showing to the image. They can actually find the correct images. And where we are going is what Matthew indicated earlier on. So that's what's coming next for us is OME and GFF. So we are also migrating a lot of the data that are currently in IDR into cloud storage. So that will be coming in the next release of IDR. So that thank you to the team, and obviously thank you to our funders.